Hello and greetings to all. Batch number 2. My name is Keerthinathan, in Hero Institute of Engineering and Technology, Computer Science Department. My project is about predicting malignant and benign cancer cells of breast tumor using supervised learning. Why this project? Uh, because breast cancer has ranked number one cancer among Indian females. Uh, so in order to address these problems, what we have done is we just try to identify what are the uh, normal data that can gather from an infected patient and a non-infected patient, and we designed a learning model from them, and we classified the infected cells and the non-infected cells as separate. So this will help us to classify the infected and the non-infected cells with non-infected and non-infected cells. So why this project? Because breast cancer is projected expected to be as high as 1,79,000 women will be affected in 2020. So in this project, what we are doing is we got this data from an institute and from an University of California. This dataset contained 569 samples of malignant and benign tumor cells. The first two columns contain the data store of a unique ID number of samples and the corresponding diagnosis, which are indicated as malignant and benign respectively as M and B. The column of 3 to 32 contained 30 real value features that have been captured from digitized images of cell nuclei, which can be used to build a model to predict whether the tumor is benign or malignant. So the object is to classify whether the breast cancer is benign or malignant and predict the recurrence or non-recurrence of malignant cases after a certain period. To achieve this, we have used a machine learning classification method to fit a function that can predict the discrete class of a new input. So in this breast cancer, from the results that we obtained from a fine needle aspiration that we got from the University of California, there we used their repository to obtain the data. So what is fine needle aspiration? It's a quick simple procedure to perform which removes some fluids or cells from a breast cell lesion or a cyst with a fine needle similar to blood sample needle. So what happened is we identified they have been separated as two, as a malignant is indicated as 1 and benign as 0. Malignant represents present and benign represents absent. So in this project, next what we are going to do is we will be analyzing the data. In the first part, we have analyzed the data completely, and we have removed anomalies to predict to have an accurate rate of predictions. So in the next stage, we have used this data, and we used exploratory data analysis. So what is exploratory data analysis? Exploratory data is a very important step which takes place after feature engineering and acquiring data. It should be done before any modeling. It's because it's very important for a data scientist to be able to understand the nature of a data without making assumptions. So we had done a exploratory data analysis. We used two methods. We used descriptive statistics and the visualization methods. In the descriptive statistics, we obtained the count, mean, standard, minimum, 25%, 50%, 75% max, those rows and all. And from these skew values, what we use the skew values from skew. What we obtained is this skew result show a positive right or a negative left skew. The values closer to 0 show less skew. From the graphs, we can see that radius mean, parameter mean, area mean, concavity mean, and concave points means are useful to predict cancer type due to the distinct grouping between malignant and benign cancer cells in these features. We can also see that the area worst and the parameter worst are also quite useful. Okay. The next what we're doing is, from this what we observe is we got 357 observation indicating the absence of cancer cells, and 212 show absence of cancer. Presence of 357 showed presence of absence of cancer cells, and 212 showed absence of presence of cancer cells. So we need to confirm this. What we did is use unimodal data visualization, and this we used the three techniques: histograms, density plots, box, and whisker plots. Okay. In visualization, what we did was first we visualized the mean using box plots. histograms. In the first we used histograms. Next we used box plots. After visualizing and analyzing, we can see that perhaps the attributes perimeter, radius, area, concavity, or compactness may have a exponential distribution. We can also see that perhaps the textural and smooth and symmetric attributes may have a Gaussian or nearly Gaussian distribution. This is interesting because many machine learning techniques assume a Gaussian unvariate distribution on the input variable. Observation. We can see the next, and we use a multimodal data visualization. What we do in multimodal data visualization is I will be separating malignant and benign cells using red and blue marks. We use scatter plots and correlation matrix methods for this. This is the correlation matrix method. In this, what we obtain is a matrix format of benign and malignant cells. The ranges between these zero are predicted as identified as malignant and non. It will not be much effective, and the cancer in the cells in the red region will be said as a dangerous region, so needs much care. So in the same is done in the scatter plots. Scatter plots, what we do is we form scatters of blue cancer data cells. So the blue indicates the malignant, which are not which will not affected or safe, and the red are considered as dangerous. In the next, we will be doing as data pre-processing. In the data processing, I export the data to help gain insight on distribution of the data as well as how the attributes correlate to each other. I identify some features of interest. I use a feature selection to raise high dimensional data, feature extraction and transformation for dimensional reduction also. So what the goal is, to predict the most features of the data and filter it, so it will enhance the predictive power of the analytics model. So first we loaded the data libraries. Next we will be encoding the labels. After that we use some assess model accuracy. In this, what is this? In this method we use some training data sets and different training methods to make this learning model using some machine learning algorithms. So what we did is first we split the available data into training sets and testing sets, as training sets as 70% and the testing sets as 30%. Train the algorithm to the first part, and made a prediction on the second part and evaluated the prediction against the expected results. Okay. So this is what we have done. Actually, next what we're going to do is we use the principal component analysis method to predict the difference in the classification and the better classification of the malignant and benign cells. So next we decide how many principal components we have to retain. From what we observed, this is the most obvious change in slope. The screen plot occurs at component 2. Which is the elbow of the screen plot. Therefore, it could be argued, based on the basis of screen plot, the first three components should be retained. Okay. This is what we observed. So next, the predictive model using support vector machine. What is support vector machine? A support vector is a learning algorithm usually used to build predictive models. So what the main goal in this is, we will be using the support vector machine to build the predictive model. What we consider some parameters in this model, in this method. There will be regulated parameter C, a choice of kernel, and a kernel specific parameter. And we have done some coding part over here, and we have done some cross validation. Next, we have used, in the next part we have used receiver operating characteristics. This is a curve that will be used to separate the cell colors, and the precision we have obtained in this is considered as 0.95%. And what we have as a receiver now we have a receiver operating character example which shows the false positive rates. Next, in the next part we will be seeing then optimizing an SVM classifier. As we already seen, SVM is a support vector machine learning model. In this we will be having a machine learning model that are parameters so that the behavior can be tuned or for a given problem. So in this we have some other coding parts and 5 cross validation. At the end, what we again obtain the precision is 0.94 percentage. We have seen there, you can see that it's separated 905 as cells that are dark, some different colors. And next, what we're doing is in this we will be classifying decision boundaries. In this method, we have completely separated the infected and the non-infected cells, and then we have clustered them together as infected and the others as non-infected. Okay. Now automating the machine learning process. For automating the machine learning process, we have used pipelines which will help overcome common problems like data leaking in your test. So we used scikit-learn and which provides a pipeline utility to help automating the machine learning workflow. After evaluating some algorithms, we separated out the validation datasets. We set up the test harness to use 10-fold cross-validation. I built 5 different models, and selected the best model. So these are the models. This is baseline, and this is what we obtained from the machine learning model. From this, you can see that the accuracy obtained is about 9.45%, 0.95%, and this is the final what we have obtained by this project. We have done some tuning in this project by using hyperparameter SVC estimator and hyperparameter KNN hyperparameters. So at the end what we have obtained is a 0.95% of precision on the classification of an infected and a non-infected cells. Right, okay, thank you. How this project ends is that in the future work we will be doing these learning models can be used to automate the treatment of cancers and for the analyzing for the specialists. Thank you.